Hi everyone, welcome back. This is lecture 7J of Useful Genetics, where we're going to continue solving problems that involve meiosis with crossovers, but now we're going to see if we can predict the numbers and proportions of the gametes of different genotypes. So here's the question we ended with in the previous lecture, where we concluded that when there were many meioses with randomly located crossovers, all of the 8 possible combinations would be seen. Now, here's another take on the question, slightly more specific. Which gamete genotype would be most likely from a single meiosis of these chromosomes with a randomly positioned crossover? And not all the possibilities are included here. So only one of these would be most likely, and that's this genotype, because this is a parental genotype. The genotype that is always present if there's only one crossover. Now, another version of the question, which gamete genotypes would be most abundant after many independent meioses, each with a randomly positioned crossover? Now I've included in the list were both of the parental genotypes, and again, they are the most likely. The other genotypes are less likely. This is a genotype that would result from a crossover in this interval. These two are the genotypes that would result from crossovers in that interval. And this is a genotype that would only be seen after 2 crossovers, which is a relatively rare event. One more. Which gamete genotypes would be most abundant after many generations of meiosis in many individuals with randomly positioned crossovers? The answer is all combinations are equally likely after many generations of crossovers, and that's because the primary consequence of crossovers is that they're gradually randomizing the combinations of alleles. And so regardless of what allele combination we started out with, if we took many generations of meiosis with many different cells, in the end we would end up with a population of individuals, of chromosomes, that had all of the possible combinations in equal frequency. Now, this is an important point, so I'm going to emphasize it in another way. If we just had one generation of meiosis, when we think about alleles that are on different chromosomes, one generation of meiosis is all that it takes to randomize the allele combinations. Those are the kinds of events that we were looking at when we did genetics problems with genes on different chromosomes that didn't involve crossing over. If the alleles are linked, if the genes are on the same chromosome, it'll take many generations to randomize the combinations. How long depends on how close the genes we're thinking about are. This applies both to genes and to SNP markers. And if the SNP markers are very close together, or the genes are very close together, it may take hundreds or thousands of generations to completely randomize the allele combinations. And the consequence of that is that we can use genome-wide association studies to exploit the linkage between SNPs and genes that cause particular effects, because those SNP differences, the different SNP alleles, have stayed linked to different alleles of the phenotype-causing gene over hundreds of generations. It's also why we can look in our genomes and detect segments of DNA that we inherited from our Neanderthal ancestors, because these segments are short enough that they haven't been randomized even after several thousand generations. We'll come back to this in a later video. So, if we were to actually predict the gamete frequencies quantitatively, we would need to know how often crossovers happen between each pair of loci. This depends, of course, on how far apart the loci are. Because each chromosome only gets one or a few crossovers, and the crossovers happen at random positions, most of the crossovers happen between genes that are far apart, and they rarely happen between genes that are close apart, close together. But we have to know the actual numbers of recombination events to predict the actual frequencies of the gametes. Now, until recently, geneticists actually exploited this relationship. They used measurements of crossover frequencies to estimate how far apart genes were, and this enabled them to construct what were called genetic maps of chromosomes, maps of the separation of genes based on measurements of recombination frequency. Now, in many organisms, geneticists are much less dependent on genetic maps because it's now cheap enough to sequence the genome and construct an actual physical map. On the next slide, I've got a couple of examples of organisms where we have both a genetic map and a physical map. On the left are maps of maize. For you Americans, that's corn. And the blue line is a genetic map based on recombination. It's not very detailed. This is how many genetic markers they had, only about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 markers on the chromosome. Here's the physical map, has many more genes indicated on it because you can identify the genes even if they're not causing a phenotypic difference. Over here, we have maps of chromosomes from yeast. Again, a physical map based on the DNA sequence and a genetic map based on recombination frequencies of markers that cause phenotypic differences that can be scored. One thing you'll notice is that although in general the markers line up quite well between the two maps, there are places where the distances are off, and there's one place where two very close markers were switched around in the genetic map. Most of these differences are because of differences in recombination frequency. Here, the genetic map showed that these markers were far apart, probably because there's lots of recombination occurring in that shorter interval. Now, as I said before, you don't need to worry about the details of differences in recombination rates. And we won't be, in this course, using genetic maps to predict recombination frequencies quantitatively. But you will be expected to be able to say, well, they will be more common in this interval than in that interval. Now, here's another problem, a slightly more complicated problem. We're considering a man who's heterozygous at two loci that are both on chromosome 7. His genotype is big M, little n, little n. And normally, in the previous problems you've seen like this, the first thing you had to worry about was, well, which alleles are on which chromosome? If we put the big M allele here and the little m allele here, then we're left wondering, well, does he have the big N allele with the big M allele, or does he have the little n allele? And vice versa. But in this problem, we're actually going to use the genotypes of the sperm to tell us which arrangement is correct, which arrangement he inherited from his parents. So here's the problem. You're given the genotypes of 100 sperm, and you have to use this information to infer which alleles were on the chromosome he inherited from his mom and which on the chromosome he inherited from his dad. And the correct answer is that the big M and little n alleles would have been together on the same homolog. And you know that because they and the little m big N combination are the most frequent. And the fact that they're the most frequent tells you that they're the parental combinations. Now, one more problem. Again, we have a man who's heterozygous for 2 genes on the same chromosome. And you're told that these genes are, you know which alleles he inherited from his father and which from his mother. And you're told that the genes are far enough apart that sometimes crossovers occur. Which of these choices describes the combination of gametes that are going to be produced by many meioses? And of course, the answer is you can't tell, because you were told only that crossovers sometimes occur. In principle, if you'd been told how often crossovers occur, you could have done a calculation that would tell you which combinations would appear. But in fact, you don't have that information, and I haven't taught you precisely how to use that information. The bottom line is, when you know they're linked, you know that the parental types will be the most common, but that's all you know. So what we've done, we've practiced predicting the numbers of frequencies of gametes with different genotypes after crossovers. We've considered genetic maps as well as physical maps, what the difference is in how they're made and that they can look slightly different. And we've used gamete genotype frequencies to work backwards to infer parental haplotypes, the combinations of alleles that were on the individual homologs in the parent. Coming up next, we're going to talk about gametes and mating before we get into doing mating problems. I hope to see you there.